Welcome everyone. This is Dr. Shruti and Alex, Assistant Professor in the Department of Biomedical Engineering. And today we'll be talking about the topic regeneration of nervous system, neural retina and optic nerve. So this will be taken from the topic Ectoderm-Derived Organs, which is taken in the course Tissue Engineering and Regenerative Medicine. So first coming to the neuron structure. So the basic structural and functional unit of the nervous system is the neuron. The neuron structure consists of a cell body which is called the soma, and the cell body contains the nucleus and the metabolic machinery, whereas the branches are the dendrites, and these receive the signals are basically your input zone, and from the dendrites the received signal is passed over to the axon. So the axons help to transmit the electrical impulses, which is similar to an output cable. And we have something called the myelin sheath. The myelinated neurons transmit signals faster than the unmyelinated ones. So the myelin acts as an insulator and forces the signals to jump between the nodes of Ranvier, which causes the saltatory conduction and thereby speeding the propagation. Unmyelinated axons generally conduct signals slowly via continuous wave like depolarization. So, for example, if a myelinated motor neuron carries information at 50 to 100 meter per second, an unmyelinated fiber carries the information around 0.5 to 2 meter per second only. So, basically, unmyelinated neurons transmit signals much slower. Towards the end of this axon, we have the synaptic terminals. So, at the synaptic terminals, they release the neurotransmitter onto the next cell. So, this is your synaptic terminal where they release the neurotransmitters, so acetylcholine, GABA, etc. And this will be received by the next neuron. And the synapse is the junction for the chemical or the electrical signaling. So there can be different types of neuron structures. So there is unipolar, wherein it has one axon, the bipolar, which is having two axon, and multipolar is basically those that have multiple branches from the cell body, and these are found in the motor neurons and the central nervous system. There are also pseudopolar, wherein there is one axon branching out, and from there it diverges into two. So basically there's only one axon, so that it's called pseudo unipolar. So coming to the structure of the nervous system, the nervous system is comprised of the central nervous system and the peripheral nervous system. So coming to the central nervous system, or the CNS, is responsible for making decisions based on the input received from the body. So it includes the brain and the spinal cord. So brain is this control center of the body, which is responsible for thought, memory, emotion, motor control, and sensory perception. It has different types of sensory inputs and processes it, and it transmits it to the different parts. Brain consists of three main parts, which is the forebrain, the midbrain, the hindbrain. The spinal cord is an extension of the brainstem down the back that helps in information transmission between the brain and the rest of the body. It is also responsible for controlling reflex actions independently of the brain in certain situations. Next, coming to the peripheral nervous system, it is responsible for connecting the CNS to the rest of the body. So the parts of the body that carry the signal to and from the CNS are the sensory afferent nerves. Then we have the motor efferent nerves. So this is what we have seen. So nervous system composed of the central and the peripheral. The CNS composed of the brain and the spinal cord. Peripheral nervous system, which consists of the somatic and the autonomic system. And in the autonomic, we have the sympathetic and the parasympathetic system. So next, coming to the axon regeneration in the central nervous system. In the adult mammalian central nervous system, in the case of any injury, that is for example a spinal cord injury, stroke, etc., the axon regeneration is very severely limited. So this leads to permanent functional deficits. So unlike the peripheral nervous system where the axons can regrow, in the case of the central nervous system, axons fail to regenerate. So what causes the failure of the neurons in the CNS to regenerate? So first, coming to the inhibitory environment. So when there is a severe or an injured axon, the myelin debris contains inhibitory proteins like the NOGO-A, which is a myelin protein that can collapse the growth cones, and MAG, which is myelin associated glycoprotein, and this can inhibit the neurite outgrowth and trigger axonal retraction. And there is OMGP, which is oligodendrocyte myelin glycoprotein, and this can block the axonal extension. So all these basically block the regrowth of the axon via ROA ROCK pathway activation. This causes a glial scar, which is caused by the reactive astrocytes, which secrete chondroitin sulfate proteoglycans, CSPGs, which creates a physical and a chemical barrier. Physical barrier, as in it blocks the cells from reaching the cells where the repair needs to be done, and this also acts as a chemical barrier. So the molecules secreted prevent the repair of the neurons too. So first we saw about the inhibitory environment. Second, there is also a lack of trophic support. So the central nervous system glial cells such as the astrocytes and the oligodendrocytes, they do not provide growth promoting factors such as the BDNF or the neurotrophin 3, etc., unlike the Schwann cells in the peripheral nervous system. So the CNS glial cells do not provide any growth promoting factors unlike the PNS. The next one is the blood brain barrier, BBB, which limits the macrophage infiltration, delaying debris clearance and prolonging the inhibition process. So the lack of trophic support hinders the axonal regeneration in the CNS. Next, coming to the neuronal intrinsic limitations. So mature neurons in the case of CNS have reduced expression of regeneration associated genes, that is RAGs, like GAP43 and STAT3. And another reason is the mTOR pathway suppression in the case of CNS neurons restricts the growth capacity of the neurons. So what are the tissue engineering strategies that can help for the nervous system repair? There are several approaches that can be used to overcome the limitations that are found in the neuronal regeneration. So some of them have been discussed here. So nerve guidance conduits. So basically you're going to provide an artificial channel. This channel will have topographical micro grooves, so basically small grooves on a material, and dimensions will be in line with the dimensions of the neurons. And combination, we can also provide biochemical cues. So the laminin provides the structural scaffolding for the neurons, and NGF basically stimulates their development. So using specific growth factors can also help in the neuronal growth and can direct the axon to grow. So we can use a combination of physical cues. So as I told you, like micro grooves or any specifically oriented fibers can be used. Okay, we can make nanofibers prepared using electrospinning, and the oriented fibers can be used for growing the neurons. You can use chemical, mechanical, biological cues that can help in the neuronal growth. So coming to the application, so they can be used for peripheral nerve injuries, spinal cord repair, and for neurodegenerative diseases, for example, Parkinson's. Advantage of this is that it avoids donor nerve grafts and it is customizable for mechanical and biological properties. The other strategies for nervous system repair include cell therapies such as use of stem cells. So these can be induced pluripotent stem cells or Schwann cells, and these can be used to repopulate the damaged areas. We can also use drug delivery, so localized release of certain neurotrophins such as BDNF or inhibitors such as chondroitinase ABC can be helpful to accelerate the neuronal regeneration. And we can also use electrical stimulation. So neurons basically transmit electrical signals. So we can give an external electrical stimulation which can help to enhance the axonal growth via bioelectronic scaffolds. So other therapeutic strategies that are under investigation are the neutralization of NOGO-A. So here we saw that NOGO-A is causing the inhibition of the axonal growth. If we neutralize the NOGO-A using certain monoclonal antibodies, or we can even use RHO-A inhibitors, so we can use specific molecules like C3 transferase. This can help in the neuronal regeneration. Another strategy involves CSPG degradation using chondroitinase ABC molecule, and there can also be trophic factor delivery. We can deliver growth factors like BDNF, GDNF via viral vectors or biomaterials, and then stem cell grafts can also be used to bridge the lesions that are in the neuronal cells, and it can provide permissive substrate wherein the neuronal regeneration can take place. So while the hurdles remain, we can use combinatorial approaches which are targeting extrinsic inhibitors along with the intrinsic growth pathways. So this can show promise for the CNS repair. So next, coming to axonal regeneration in injured spinal cords. So in general, if we look at the dorsal root ganglia sensory pathway, the pseudo-unipolar DRG neurons, they lack dendrites. Their peripheral branches innervate the skin, and the muscles, while the central branches, which is composed of the dorsal column axons, they enter the spinal cord's dorsal horn. Synapse is where the dorsal column axons connect with the second order neurons in the spinal cord, which ascend via the dorsal column medial lemniscus pathway, and this carries the touch or the proprioception. And then decussation is when the axons cross at the medulla and relay the information via the thalamus, which is a third order neurons, and project the information to the somatosensory cortex for perception. This is the general sensory pathway that is found in the spinal cord. So, what are the regeneration challenges? So, here also to some extent the glial scars, which has the CSPGs, and the myelin inhibitors like NOGO-A can block the regrowth of the axon. Therapeutic targets look at degradation of the CSPGs with chondroitinase ABC, neutralize NOGO-A with the antibodies, or combine scaffolds and growth factors like neurotrophin 3 to bridge the lesions. So we had seen this even in the central nervous system. And we can also look at combinatorial therapies for future direction. So, we can look at specific nerve conduits in combination with stem cells and electrical stimulation, which can help in the functional recovery of the neuronal cells. This diagram basically shows when there is a spinal cord injury, what are the different factors that affect the regeneration. So, they are diminished intrinsic neuronal capacities, so limited Schwann cell remyelination, inhibitory molecules that we have discussed, glial scars, restricted regenerative cell migration, plasticity, which is less for the neurons, and compromised vascular supply. So, the spinal cord and the brain are part of the central nervous system. The limited regenerative capacity is found in general in the central nervous system. So, we've discussed the regeneration ability of the CNS and what are the tissue engineering strategies we can use to overcome the low regeneration capability of the CNS. Next, we'll be looking at other neurons. So, first, we'll be looking at axonal regeneration in something called the neural retina. Neural retina is a light sensitive tissue which is found at the back of the eye, and this helps to convert the light into neural signals. So, the rods and cones convert the light into the neural signals. So, the retinal ganglion cells have several regeneration challenges. So, they fail to regenerate axons after an optic nerve injury or glaucoma, and this can cause the permanent vision loss. So, what are the key barriers here? So, we have weak intrinsic growth capacity in mature RGCs, inhibitory signals like the NOGO and CSVGs in the optic nerve, and also poor RGC survival post injury due to the neurotrophin deprivation. So, growth factors needed for the nerves are deprived after an injury. So, these are the reasons why the axonal regeneration in the neural retina is restricted. So, what are the strategies that we can use to overcome these difficulties? We can look at activating the RGC growth state. So, we can upregulate pro-regenerative genes such as the KLF4, mTOR, STAT3 via viral vectors or small molecules. So, these regenerative genes, when they are upregulated, it will help to fix the damaged neurons. And also we can look at counteracting the inhibition or the inhibitory molecules. So, blocking molecules that are responsible for the inhibition of the neuronal growth like NOGO, PTP, sigma receptors, or digesting the CSPGs, example using chondroitinase ABC, can be helpful for counteracting the inhibition. Neuroprotection. So, we can deliver certain neurotrophic factors like BDNF, CNTF, or GDNF to enhance the RGC survival rate. So, what are some of the recent progresses that are done in this field? So, animal studies have shown that partial axonal regrowth around 5 to 10 millimeter was observed when they combined optic nerve conditioning, wherein they have used inflammatory priming along with gene therapy. They have used OC1 overexpression, and they have also used a scaffold with growth permissive substrates such as laminin, fibronectin, etc. What is inflammatory priming? In the case of a lens injury or an intraocular inflammation, this activates macrophages, so increasing the intrinsic growth capacity of the RGC and overcoming the myelin inhibition can help to prime the inflammatory cells. In the case of OC1 overexpression, this basically reprograms the mature RGC to a regenerative state, and it also helps to upregulate growth associated genes and enhances the axonal extension. molecules like laminin and fibronectin provide a physical bridge and adhesive cues that can guide regrowing the axons across the injury site by bypassing the inhibitory glial scars. What are the hurdles that remain in the regeneration of the neural retina? The regenerative axons must reconnect accurately to the lateral geniculate nucleus targets. So the axonal guidance must make sure that right axon must connect to the right nucleus. It is not sufficient that the conduit is provided. Appropriate connections must also be guaranteed. The next hurdle is about the functional integration, wherein the new synapses must restore the visual signal transmission. So phase 1 clinical trials testing the neurotrophic factors such as CNTF implants are underway, and combinatorial approaches using gene therapy and rehabilitation may reach the clinic within 5 to 10 years. Next coming to the regeneration in the case of optical nerve. So the optical nerve carries the information from the retina towards the brain. So in case of any damage in the optical nerve, so we can look at gene therapy. So this targets intrinsic growth barriers by delivering pro-regenerative genes such as mTOR, KLF4, or silencing the inhibitors such as P10 and SOC-S3 via adeno-associated vectors. So recent advances involve CRISPR-edited RGCs, which show enhanced axonal growth in rodent models. So next, we can also use stem cell therapy, wherein induced pluripotent stem cells, wherein stem cells are derived from adult cells such as skin cells, fat cells, and they are reprogrammed to become the stem cell-like characteristics, and these are derived from the RGCs, and they can integrate into the retina and help to extend the axons. These have been done in the mouse studies. These cells have shown neuroprotective properties. So, for example, mesenchymal stem cells secrete BDNF and CNTF to rescue the dying RGCs. So, the challenge here is that ensuring the synaptic rewiring to the accurate LGN nucleus targets is something that is still underway. Next, we can also use scaffolds or biomaterials. In specific, as I mentioned, electrospun nanofibers. Using rotating mandrels, it is possible to obtain aligned PCL gelatin nanofibers that can help to guide axons with topographical and chemical cues such as laminin and NGF. So this can help in the regrowth of the neurons. So apart from this, injectable hydrogels help to deliver growth factors while bridging any lesions. And 3D printed bridges with microchannels help to improve axonal directionality. These are some of the strategies that are used for the regeneration of the optical nerves. So critical barriers that are there in the optical nerve regeneration are, first is the distance. So in the case of rodent models that have been studied in lab, the regeneration that was needed was around only 5 to 10 millimeter. Whereas in the case of human optical nerve regeneration, this requires up to 50 mm of regeneration. So this is one of the major critical barriers that are seen in the human optic nerve regeneration. And the second major issue is the timing. So chronic injuries accumulate scar tissue. So with time, there are a lot of scar tissue that is built up, and this can reduce the intervention efficacy. So if treatment strategies are done at an earlier period, the possibility of regeneration is much higher, because over time the scar tissues can build and the possibility of any intervention is limited. The clinical pipeline that exists now are phase 1, phase 2 trials which are testing CNTF implants and adeno-associated viral 2 BDNF delivery vectors. And there's also biomaterial scaffolds which are entering preclinical testing for chromatic optic neuropathy. Here it shows a site of optic nerve damage, and in this case an allogenic transplantation. So basically the retinal ganglion cells are taken from a healthy eyes into the host eyes, which may represent a viable strategy for curing irreversible form of blindness. RGCs are taken or transplanted from a donor and receive the intrinsic signals, and this will drive the initial neuroid and the axonal outgrowth. Once the neuroid and axonal growth happens, the complete integration into the retina and the axon grows into the optic nerve. So thereby it is possible to regenerate the optic nerve and thereby help in fixing the optic nerve damage. So with this, we are ending this session. Thank you.